Alright, so I want to start today's lesson off with a little bit of help for the biotechnology stuff. You are going to be working on two different labs, and today's lesson is going to help you out with your pGlo lab. So with our biotech stuff, we are entering a new world of understanding genetics and what we can do with that then. Can we make animals glow in the dark? Can we make proteins for a human body? Can we figure out suspect's blood and blood that's found at a crime scene to either convict somebody of doing a crime or to set them free? So very interesting things that we're going to be learning about in the next couple days. So let's just talk a little bit about the human genome, and I just kind of put some letters up here of DNA just to kind of keep in mind we've got 3.2 billion bases in our genome. And then we even add another layer to that in how we organize those bases into chromosomes. So we've got a pretty complex thing to work on. So humans weren't necessarily our first test subjects in biotechnology. Instead, we work with some organisms that are a little bit simpler to get started, and we hope to be able to take some of those things that we learn and apply them to the human genome. So let's talk a little bit about genetic engineering. As basic as it gets is it's how can you take DNA and manipulate it. And if you're going to engineer DNA or genes or organisms, then you need a set of tools. And that's why we did the pGlo lab, because it gives you an example of a set of tools to use. The first is the life form that we find easiest to use, and that's bacteria. And this slide just shows some very basic stuff about bacteria that we've covered. Prokaryotes, single-celled, really fast growth. So they're very simple to use, and it's something that we can do in a lab and overnight have an entire colony. And then if we look into their genetics, they have a very simple singular chromosome. You'll learn today, Friday, about histone proteins and why that can cause some complexities. Histone proteins are found in plants and animals. Bacteria do not have them, though. And we're looking at a significantly less amount of base pairs, only about 4 million, only 4 million. And that gives you about 4,300 genes then. One of the ways that we talked about how bacteria become genetically diverse is through a process called transformation. And what they will do is actually take in bits of chromosomes from other bacteria. And that bits of chromosomes then gets incorporated into their own chromosomes. So it's a way for them to express new genes or another form of recombination. And so they would need to do this because when we talked about bacteria, they have to be able to rapidly change along with their environment. They don't have some of the things that eukaryotes have in place with homeostasis. And Griffin's experiment, this is exactly what was happening here. He was able to take bacteria that were harmless and transform them into bacteria that ended up killing his mice. So the other thing that we can't leave out when we talk about bacteria are the plasmids. These carry somewhere around 2 to 3 genes. They replicate on their own. That's kind of a key point here. And it's also something that we've learned where antibiotic resistance is at. Now that is where you definitely want to focus on for your pGlo lab. There's something here going on with antibiotic resistance, and see if you can apply that to some of your results. You can also import some of these plasmids from the environment. Just like we talked about, bacteria will take in genes from other bacteria. If there's plasmids in this bacterial environment, they can bring those in, and that's exactly what we did. We had plasmids that we brought into our bacterial cells. So this is kind of what we did for our procedure-wise. We had a plasmid that we introduced into our bacteria. We cut that plasmid and incorporated a gene from another organism, and then allowed that bacteria to grow. And so why would we want to do that? Well, we can take genes, like genes for insulin or human growth hormone or lactase, we can use ligase, which is an enzyme, to put that into our bacterial DNA, and then that gene would be expressed. So just another picture to show what's going on. So these scissors that I have as a graphic, they are not actually little scissors floating around in bacteria, but they are enzymes, and they're called restriction enzymes. And these are naturally present in the bacteria. It's actually part of their defense mechanism, because bacteria can be invaded by viruses. And what that virus will do is it will inject its genetic material into the bacteria. The bacteria is always looking for new genes to incorporate into its genome to allow it to survive and possibly reproduce. However, what it doesn't know is that this is bad genes. And what will happen is that viral DNA will be transcribed and translated and made into more viruses, ultimately killing the cell. So the way that a bacteria will get around this is it's got restriction enzymes that are present, and it will very restricted. It doesn't just cut up all DNA, but it does look for specific sequences and then cut that up. And so if you cut up the viral DNA, then that is a protection against the cell eventually being taken over by a virus. So these restriction enzymes work in a particular sequence, and that's what I want to show you. Just take a look at these terms and sentences that are coming across your screen. What do you notice? And if you said they read the same left to right as they do right to left, you are correct. It's called a palindrome. And that's exactly what a restriction enzyme does. It will read the same 3 to 5 or 5 to 3, make little cuts, and then what you end up with is something called sticky ends. They're the ends that are no longer bonded, but we can use those sticky ends, and you'll see that here in a second. So here's my example of DNA in white, and the area where a restriction enzyme will cut is in red, and you'll notice that it cuts between the G and the A, no matter which direction we're reading. And when that cut is made, it produces sticky ends. We did this with our plasmid DNA that you cut. So I've got the white, which is the gene that I want. I cut the chromosome where I want to add that gene with the same enzymes so that I produce the same sticky ends, and then you can see how they line up when they are combined together. Now another underlying factor in this is that how these codes are read and how they're translated into proteins is universal. All organisms here on our Earth use these 20 amino acids to make up their proteins, so it doesn't matter if you are a plant, an animal, a protist, a fungus, or a bacteria, the language is the same. So after we have put the instructions for making that trait into the bacteria, we said we have a recombinant plasmid. You can grow that bacteria, make lots and lots of copies of that plasmid, because remember, those are self-replicating. And you should have a transformed phenotype, or a new protein. And you can actually take and harvest that protein, and that's what we do with humulin. And we use that for insulin. So other ways of looking at this genetic engineering perspective, we can genetically modify organisms, GMOs, and you hear a lot of this in the news when it comes to certain crops. One of those being corn, genetically modified corn. We can take a gene from Bt, which is a bacteria that naturally exists in soil, and put that into corn. Well, what happens is when any type of worm, like the corn borer, eats corn that has Bt in it, that Bt basically turns all the insides of these caterpillars to mush. So it's like having an insecticide built in. Now, what does that do when humans eat this corn? Well, Bt does not do anything to our insides. It's something that targets specific proteins in the caterpillar. But things like Bt corn, and there's another product called Roundup Ready soybeans, what that does is the soybeans have a gene inserted into them that gives them resistance to the herbicide Roundup. So a farmer will plant the soybeans, and once the weeds start sprouting, we'll go and spray everything with an herbicide. While the Roundup Ready soybeans, they are not harmed by how the chemicals work in that herbicide, but everything else is killed. So we do have some problems where we're seeing weeds who are becoming Roundup resistant. And we're not entirely sure, there's not enough research out there to tell us when we genetically modify our food, how does that impact us in the food chain. There was a recall of Taco Bell products that used BT corn in the 1990s, and that kind of sparked a lot of the controversy with genetically modified foods, and they are not approved for human consumption, but we do use them in things that would have corn-based or soy-based products in them, things like ethanol, some beauty products that have soy in them. Two other examples I have, something called fish berries. This would help to extend the growing season for agriculture because they are strawberries that have anti-freezing genes in them, actually from fish. Another one would just improve quality of food. Rice, which is often used to help feed poor countries who cannot produce their own amount of food just for whatever reason, drought, disputes, things like that. If we put vitamin A and other nutrients into the rice, you would basically be able to eat a very simple food and get all your nutritional requirements. So definitely some interesting things to think about, some things that you are going to see in the future and have to be able to make some informed decisions on. All right, hopefully that helped you for your pGlo, and then our next section that we're going to look at is using these restriction enzymes and getting DNA fingerprints.